Good afternoon, everyone, and thanks for tuning in to today's presentation. Today, I'm excited to highlight the application of humanized cytokine models to assess the in vivo efficacy of antibody therapeutics. I'll start by providing a very brief introduction to our company and the services we provide. Biocitogen was founded in 2008 in Worcester, Massachusetts. We initially focused on services in the gene editing arena, where we've developed proprietary methods that improve efficiency, even for very large insertions. Harnessing these technologies, our company has since created a vast array of humanized animal models to support the research endeavors of our clients. After growing into one of the largest animal model suppliers in China, we opened our branch in the greater Boston area in 2018 to expand our services. Using an advanced chromosome engineering technology, we recently introduced our fully human antibody mouse, RenMab, which contains the entire human immunoglobulin variable domain, including the surrounding non-coding regulatory regions in situ. The use of this mouse and its related platforms are powerful models for fully human antibody discovery. If you'd like to hear more about how these models can advance your antibody discovery application, please come visit our virtual booth and connect with us there. Finally, we perform a variety of in vitro and in vivo preclinical services, which I will also discuss further with you all today. So altogether, the ability of our company to integrate these technologies makes us a one-stop solution that can help take your project from target to clinical development. For the rest of today's presentation, I'll provide a brief overview of what we know about the cytokine production and its involvement in immune dysregulation. I'll then discuss some of the general challenges that have been faced in bringing some of these therapies to clinic and how humanized mice can help to bridge this gap. I'll provide specific case studies in the context of 3 different experimental models for 2 cytokine signaling pathways, IL-4 and IL-17. Finally, I'll put our work into the context of FcRn biology, where ideally we'd like to maximize the levels of therapeutic IgG recycling, but minimize levels of pathologic IgG in the context of other immune disorders. So as we all know, cytokines are important growth factors that dictate cellular responses during infection, inflammation, and cancer. They're produced by diverse cell types and ultimately regulate maturation, growth, and responsiveness to stimuli. Depending on the types of antigens that are sensed, cytokines ultimately control the T cell subsets that expand during the resultant immune response. There are a few diseases known to be generally associated with the Th2 response, which is a specialized response that normally functions to maintain barrier immunity against pathogens in our environment. However, these pathways can become inappropriately active in response to inciting allergens. For example, in atopic dermatitis, immune activation causes immune cell infiltrate and bright red skin that is thickened and scaled in appearance, yet the barrier is impaired from the damage. In the case of asthma, inappropriate Th2 response in the airway can cause excess mucus production, smooth muscle contractility, and airway obstruction. Interestingly, these diseases and other Th2 diseases, including allergic rhinitis, food allergies, and conjunctivitis, are also strongly associated with one another. Therefore, targeting the common inflammatory mediators of Th2 disease is of significant clinical benefit. The central drivers of Th2 disease in response to allergens and environmental stimuli are the inflammatory cytokines IL-4, IL-5, and IL-13. In particular, IL-4 can act early in the process of Th0 commitment to a Th2 phenotype. IL-4 can be sensed by both immune and tissue cells and mediates multiple steps of the pathogenic cascade. And mice that overexpress IL-4 actually develop spontaneous atopic dermatitis, indicating that this pathway is of significant clinical interest. So to date, there have been several therapies targeting multiple Th2 cytokines, and many of these therapies have had limited success in specific pathologies. Thus far, dupilumab, which targets IL-4 receptor alpha, is the first launch drug targeting IL-4 that has demonstrated success across atopic dermatitis, asthma, and allergic rhinitis. Despite these advances, we still need to develop more therapeutics to have a broader reach. So how are we going to best approach validating novel therapeutics and those currently in development? Well, the use of humanized mice is an ideal preclinical model for this application. So some of the advantages of using humanized mice for cytokine targeting is that the animal has an intact immune system, as opposed to immunodeficient model engrafted with human PBMCs, which might not develop properly. Because these studies are in vivo, they allow for mechanistic studies to confirm that the therapeutic candidates are performing their function as intended. And because we're working with mice and not non-human primates, the studies are lower cost and it's much more feasible to study larger cohorts. The main disadvantage is that the mouse and human immune system are not completely identical, so in some cases, disease may not be completely recapitulated. For proper cytokine receptor signaling, in many cases, we may need to create doubly humanized mice for both cytokine and receptor for intact function and characterization. An additional challenge in the therapeutic targeting and humanizing of cytokine strains is the shared receptor domains and homology that normally enable some crosstalk and/or redundancies between the pathways. Luckily, at Biocitogen, we put in immense efforts to properly design, create, and validate models to ensure maximum applicability for our strains and thus maximum translatability. In the case of targeting IL-4 signaling in Th2 disease, we can see here, for example, that IL-4 shares both receptor chains with other cytokines. So you can see that IL-4 shares the common gamma receptor with IL-2, IL-7, IL-9, and IL-21. And shares the IL-4 receptor alpha chain with IL-13. Since both IL-13 and IL-4 play roles in the Th2 disease, which, as I mentioned earlier, targeting IL-4 alpha in addition to IL-4 alone may actually be of greater clinical benefit. So to ensure proper cytokine signaling, we employed a dual targeting strategy to create a humanized IL-4 receptor alpha model. So while the soluble cytokine has been completely replaced by human sequences, as shown here in the pink, importantly, the intracellular portion of IL-4 receptor alpha, which is shown here as the black exons, remains unchanged so that downstream signaling is theoretically unaffected by replacement of the extracellular domain. For every model we make at Biocytogen, we want to ensure that the alterations do not affect the overall health and immune function. And as you can see, there's no significant changes in the overall frequency of leukocyte and lymphocyte populations as assessed by flow cytometric analysis of splenocytes. To determine whether the humanized models could indeed produce functional IL-4, splenocytes from wild type and homozygous IL-4 receptor alpha humanized mice were isolated, stimulated, and analyzed by ELISA and flow cytometry. As you can see, mouse IL-4 is detected in wild type mice as expected. However, you can see that human IL-4 is induced exclusively in the homozygous humanized IL-4 receptor alpha mice. Similarly, using flow cytometry, we can see that while murine IL-4 receptor alpha is detected in stimulated splenocytes, as shown here, human IL-4 receptor alpha, but not mouse IL-4 receptor alpha, is detected in the humanized mice. To test the functionality of the secreted IL-4, we examined IgE production by ELISA and observed that ex vivo stimulation of splenocytes with mouse IL-4, as shown here, cannot induce IgG in the humanized mice. In contrast, induction with human IL-4, as shown here, can nicely induce IgE expression in the IL-4 receptor alpha humanized mice. In addition, incubation of splenocytes with dupilumab, which as a reminder blocks IL-4 receptor alpha, resulted in a substantial decrease in IgE production, further indicating that this pathway is functional in the humanized mice. So for the first in vivo case study, we employed a model of atopic dermatitis induction and treatment. This is achieved by oxazolone sensitization by injection in the ear, and subsequent challenges as shown here on this timeline. In some groups, dupilumab was also administered to test the efficacy in preventing atopic dermatitis-like symptoms. So as you can see, the ear thickness was increased in all treated mice compared with the vehicle control. However, upon histologic analysis of the ears, you can visibly see a phenotypic improvement when comparing ears for mice treating with increasing doses of dupilumab compared to the isotype control. This was quantified by the assessment of eosinophil infiltration, where increasing doses of dupilumab was able to reduce the infiltration score, as shown here on a scale of 0 to 2. Importantly, increasing doses of dupilumab treatment was also associated with a reduction in IgE production, as shown here, demonstrating its effectiveness in preventing disease severity. So our next case study with humanized IL-4 receptor alpha mice is assessing dupilumab efficacy in treating an animal model of asthma. So for these experiments, mice were first pre-sensitized, as shown here, and then further challenged with antigens on the timeline as shown. Dupilumab treatment was given intermittently, also as shown. So after 26 days, the airway histology of the animals was assessed, and inflammatory cell infiltrate is observed with both isotype and dupilumab treatment. However, the numbers of CD45 T cells, as well as the numbers and the percentages of eosinophils, which were enumerated in the bronchial fluid, was significantly improved with dupilumab treatment when compared to the isotype control, which was shown in red. Importantly, IgE production in both isolated serum and bronchial fluid was substantially reduced with dupilumab treatment. So altogether, these data indicate that the humanized IL-4 receptor model is able to recapitulate Th2 disease and provides a robust platform for testing the efficacy of new therapeutics. So for the final case study, I'll shift focus to the Th17 response, which is driven and perpetuated by several cytokines, including IL-17. So IL-17A and IL-17F are homologous cytokines produced by activated Th17 cells, but it is becoming more appreciated that other cell types can also contribute, including gamma delta T cells, CD8 T cells, NK cells, neutrophils, and mast cells. IL-17A and F can synergize to signal through IL-17R, and high levels of both can be dysregulated in the context of several autoimmune diseases. So to help block IL-17 signaling in these disease contexts, there have been several antibodies developed to target IL-17. In the case of psoriasis, which I will focus on for the next case study, IL-17 ultimately functions to recruit inflammatory cells into the skin, which causes the keratinocytes to become activated and proliferate. So to examine the efficacy of these drugs and new antibodies in the pipeline to target IL-17, we've developed a dual humanized model as shown with both IL-17A and IL-17F humanized. IL-17A is completely humanized, as shown here in pink, while IL-17F maintains a small portion of the mouse protein for optimized expression purposes. As you'll see in a couple of slides, in this case, humanization of the IL-17 receptor wasn't necessary to maintain intact downstream signaling. So to confirm secretion of IL-17F and IL-17A, different strategies were employed. So for IL-17F, CD4 T cells were sorted from splenocytes of wild-type and heterozygous IL-17A and IL-17F humanized mice. In this case, these were heterozygous mice. So you can see a little bit of mouse IL-17F production in the humanized model. These cells were stimulated into Th17 cells. Again, so you can see that mouse IL-17F was detectable in supernatants from wild-type mice and heterozygous. Conversely, you can see that human IL-17F is exclusively detectable in the heterozygous humanized mice, but not the wild-type C57 black 6 mice. For the IL17A analysis, a similar strategy was employed. In this case, the mice were stimulated with anti-CD3 and CD28 in vivo and analyzed by ELISA. Again, the same scenario where you can see a little bit of mouse IL17A maintained in the heterozygous humanized mice, whereas human IL17A is exclusively produced in the humanized model. Furthermore, these analyses allowed for confirmation of the normal distribution of immune cell components in the spleen, and shown on the right is the quantification of T cell subsets. So as you can see, there's no real significant alteration in the T cell subsets in these humanized mice. To test the efficacy of antibody candidates targeting the signaling pathway in vivo, an experimental model of psoriasis was used, where imiquimod cream was applied to induce symptoms of the disease repeatedly over a period of 6 days. At the same time, bimekizumab, which is an anti-IL17 therapeutic, was intermittently injected at low and high doses, as you can see in the timetable. So as you can see from the blue and red curves, the anti-IL17 treated mice have an overall significant reduction in both the erythema score and the scaling score when compared to those mice not treated with the IL17 antibody, which is shown here in green. And you can really see the phenotypic difference on the back skin of these mice treated with the anti-IL17 therapy. Histologic analysis of the back skin of these mice also revealed that the higher doses of anti-IL17 treatment significantly improved metrics of the disease and the overall phenotypic score. You can see the epidermal thickness is significantly improved in the mice treated with high doses, as well as the overall histology score. So therefore, these humanized IL17A and IL17F mice are a validated, valuable tool for in vivo efficacy of anti-human IL17 antibodies. So in addition to these cytokine models, I've summarized the status of our other humanized cytokine and autoimmune models at Biocitogen, which are sorted by known target and as well as possible targets. In addition to these models, we have over 100 humanized models available on our website with several in development. So please come to our booth to learn more about the models we offer and how we can help support your applications. So in the final few minutes, I would like to briefly introduce the humanized FcRn model we're working on for future experimental targeting and validation studies. So as you can see, the entire coding region of mouse FcRn has been replaced, so exons 2 to 7 shown here in pink, by the human exon, and expression of the human gene is confirmed here by real-time PCR. And thus far, preliminary testing of the model indicates that humanized FcRn groups, as shown here in green and purple, do not exhibit substantially different kinetics in the persistence of IgG after antibody treatment. So we predict that these mice in the coming months will hopefully be an effective tool for preclinical pharmacokinetic evaluation of your FcRn targeted therapeutic antibodies in development. In addition to the humanized mice for testing therapeutic targets, as I briefly mentioned before, we also have a humanized immunoglobulin mouse model for robust therapeutic, fully human antibody discovery. So for those interested in pursuing antibody discovery with our platform, I wanted to mention that our team has also assembled a knockout library with highly homologous or difficult targets knocked out. So you can go on our website and search the library for your target of interest and see the status of the KO mouse models, including FcRn, which you can see here is available at the F0 stage. So using these KO models means you can guarantee the best and most robust generation of fully human antibodies to your target. So if companies are interested in partnership with us with this line, please do let us know. So to summarize, I hope I've convinced you that humanized mouse models are immensely powerful tools. They provide a convenient, robust preclinical platform for testing the pharmacodynamic properties, pharmacokinetic properties, and efficacy of fully human or humanized antibodies or other therapeutics, and are widely applicable to many disease pathologies. Specifically, Biocitogen's humanized cytokine mice can be used to demonstrate efficacy of leading drugs targeting IL-4 and IL-17 signaling And in particular, the IL-4, IL-4 receptor alpha model has helped advance several drug candidates to IND in China. These case studies are just an example selected from the many humanized models we have to help illustrate the broad range of applications possible, including antibody discovery and mechanistic insights. Thank you all for your attention, and I'm happy to take questions.